do work for the Marine Corps. That's not me. Okay, that's Colonel Hart. And I will talk a little bit, well, I'll talk a fair bit about Head Start, which is the topic of this article. But I also want to talk a little bit about some of the stuff that got me to that point. So, and also about some of the stuff I've done overseas. So I'm hoping that some of that information and pictures and things will also be interesting for you. Also, since I do work for the federal government, I received a document that says I must make sure I tell you I'm not here today representing the government. I'm here on my own personal basis. That's part of what I deal with in my job. Okay, so what I want to do is get to Head Start, but tell you how I got to that point. And I'll start off, next slide. I'll start off first talking about my earlier work with the Desert Tortoise, a couple of different projects. And then while I was doing some postdoc work, I also had a chance to go to Uzbekistan and work on the Central Asian tortoise. I know a lot of folks here call them Russian tortoises. But while I was there, a guy from Russia said, "Why do you call them Russians? They're not in Russia." So anyway, so I tend to call them Central Asian tortoises. I also spent some time in South Africa, so I'll talk a little bit about that. And that was actually a fabulous experience, and I still have colleagues that I work with. And then I'll come back, sort of full circle, coming back to the stuff working on the desert tortoise we have here in the Mojave Desert. Okay, so for my dissertation, I started working on the Mojave Desert tortoise, and I was working on a field site near Needles, right about here. And the reason why I chose this site was a couple-fold. One, there was already a researcher there doing some studies, and it was a very good field site with a lot of tortoises. Since then, that population has declined. Excuse me. But one of the things about the desert tortoise is that it lives in a desert environment. Next slide, please. Now, deserts are defined by a couple factors. Definitely, they have low precipitation, low rainfall. But it's not just that. It's also variable rainfall and unpredictable rainfall. Now, we've got a species here, desert tortoise, that has to deal with all those things. It has to learn how to endure harsh temperatures, harsh droughts where there's no food to eat, or essentially no food to eat. And then go to the altitude extreme where we have El Nino events, and there's 1,000 times as much food as during those drought years. Now, the researcher at that site, Fred Turner, he found that the female tortoises, they were producing eggs almost every year, and the variability in egg production was not closely related to the amount of food or water or the rainfall. So he wanted to know why, but he didn't have the particular skill set to answer that question. My professor and I were in the process of developing projects that would help address that. Now, Fred, when he was doing his studies, he thought these females may be able to produce eggs every year if they are storing up resources over time. So that by the time they enter their winter dormancy, 5 or 6 months, they've already got resources that help them reproduce the following spring. So that was essentially the question I was about to ask with my dissertation. Do the females store up energy or other resources before winter and use them the ensuing spring to help them produce eggs? Short answer is yes. Long answer is a little more complicated, but in essence, yes. I'll just quickly highlight the yellow. That's their ability to store, that's the rates at which they were storing fat or lipids. The red, so if it's positive, they're storing, if it's negative, they're burning it up, they're using those lipids for something. On the opposite side, there's red. Now that's to indicate the non-lipid component of the body. And that is most likely going to be protein, but it could also be carbohydrate. The green is metabolic rate. how much energy have they invested in activity, mating, foraging, fighting, sleeping, basking, all those sort of things. And you can see that varies a lot. And then in orange, here and here, is them actually committing resources to producing eggs. So this was a 2-year study, started at the end of a reproductive season right here, and followed through this first reproductive season in 88, and then continued for a second year through the reproductive season in 1989. On top of that, the first year was a year that had relatively average rainfall. But the second year was the driest year recorded for the site, at least at the time. So like in that earlier picture, there was essentially no food for these tortoises to eat. I mean, they came out, they found a few dry twigs and little odds and ends, and they ate that. But there wasn't really any food. But they still managed to produce eggs. Also similar to what Fred had found earlier. Well, how do they do this? One of the things is they are expending a lot of energy in terms of energy metabolism in the wet year. But in the dry year, they reduce their activity. They come up and they see there's no food to eat. So after a while, they just give up and say, okay, I'm going back down. And if you look right here, compare this to here, you see this is 90% less. And here, compared to this, it's 75% less. So they're reducing the amount of energy they're spending on activity and types of costs associated with activity. What they can do then is they can use that energy for other purposes, such as producing eggs. So the tortoises, and I'm sure that many of you have a basic feeling for this as well, they're very, very opportunistic, and that's where this concept of homeostasis comes in. When those resources are available, they'll drink. They'll come out and drink, and when the storm comes through, they know that they'll actually take bases in the sand that the water's pulled up. They're very opportunistic. When it's there, they'll draw upon it. Including, well, for instance, right here, there's a summer rainfall. They're rehydrated, and all of a sudden, they can eat all the dry grass available. When springtime rolls around, they can also eat the green vegetation, and that helps them store protein. So, get back to this idea of homeostasis. You and I maintain our body temperature within a very narrow limit. We regulate our blood glucose within very narrow limits. We regulate our concentration of our body fluids and other things within narrow limits. So do other mammals, and so do birds. Reptiles don't do that, for the most part. Reptiles don't regulate within these very, very strict limits. They allow things to fluctuate. And so it's sort of a homeostasis. They do regulate, but it's much more relaxed. So their body temperature can go way down and come back up. Their body lipids can go way down, but they can build them back up as well. Same thing with their water. So this homeostasis, they do regulate, but in much broader intervals than what birds and mammals do. And this helps them survive. Remember, these guys are in a desert where rainfall is already low, but it's also variable and unpredictable. And so is the food that's available to them. So one of the mechanisms that they use to survive, these guys are out there living in the desert 50 or more years. How can they do that with all this variability? It's their physiology that helps keep them able to do that. Now, as I mentioned also, the second year was a drought year. Some of the females also still produced eggs. Extreme drought year, but they still produced eggs. They also, in the process, forfeited some of their body reserves. Now, why would a female, this is a question we can talk about later, but I'm posing it now. Why would a female in the middle of a desert, in one of the driest years they've probably ever experienced, forfeit body water and body protein to help produce eggs? Okay, so think about that. Now, as I mentioned, the females, they change their body composition a lot. Body lipids, body body protein and things like that. But in those earlier slides, what I showed you was not, it was lipids, but the other component was not protein per se. It was just the non-lipid component of the body. And we think that's protein, but we couldn't prove it with those data that we had. So I then collaborated with Olaf Optedal at the Smithsonian Institution, and we worked out at that Tortoise Center that used to exist out in Las Vegas. We did some really great work. One of them was we took females and we fed them diets with different protein contents to try to see if the protein content was going to influence their ability to produce eggs. So we fed them diets with different protein contents. We measured when they ovulated and how many eggs they were producing. And we did that for about 18 months. So from the beginning of 96, as they came out of hibernation, we fed them these diets. And then we measured when they produced eggs. We carried it through for some of those animals, not all 24. We carried it through for another year, for another whole 12 months. So we also looked at nitrogen intake and or protein intake and reproduction in the spring of 97. Now in that spring period, whether it was 96 or 97, the amount of eggs they produced was not at all correlated to how much protein that they consumed. So the immediate amount of protein was not correlated, or the reproduction was not correlated to that protein intake. However, what about long term? Spring, next one please. Spring of 1997, the number of eggs they produced was strongly correlated to how much nitrogen or protein that they consumed over the previous 9 to 12 months. So it's the long-term consumption of nitrogen that's influencing their ability to produce eggs. Next slide. In fact, if we take a look, this is just a time frame, again, like the 18 months of time. The first year and second year of egg production. The egg production here was related mostly to nitrogen consumed over these intervals, all the way back to the previous year, with 60% of that nitrogen or protein consumed previous spring. So most of the protein that they're consuming that influences their ability to reproduce is consumed the previous year. So there's a lag effect. Next slide. Why? Well, we think it's vitellogenesis, and we know, we've done ultrasound work. Females are developing their follicles in those ovaries, mostly in the summertime and the fall of the previous year. When they go down for the winter, these follicles, the big ones here, those are full size. Before they go down for winter, those follicles become the egg yolk the next spring. So when they come out the next spring, they may not even eat before they ovulate, because they've already got most of it already ready. Those yolks also have 90% of the protein in the egg. So we think the process that's slow, and the reason why these females were producing in relation to food consumed the previous year is because that food consumed the previous year is what's enabling them to develop these follicles. That's the time period when they're getting ready for next year's reproduction. Now, it's not quite that simple. We can talk a little bit more about it. But there is a little bit, like if there's a little bit of an effect of the current year food availability. If it's an El Nino year, there might be a lot of food, and females might be able to squeeze in a second clutch. So the current year might be able to influence reproduction, but it's mostly what they've got ready before that. Next slide. Okay, so let's take a little field trip. Let's go over to Uzbekistan and look at another species of tortoise, desert tortoise. Again, Russian or Central Asian tortoise. This is roughly what their range is, not in Russia. And I worked with a couple of French colleagues, Frédéric Lagarde and Xavier Bonnet, as well as Dr. Nagy from UCLA. Here, just outside of Bukhara in Uzbekistan. Next slide. So, this species of desert tortoise has a few sort of distinct traits about it that make it much more, well, not more interesting, but make it interesting in comparison to the Mojave desert tortoise. So so we wanted to identify some of those things. So we looked at diet and activity, how far they moved, hormones, and then I also, again, was looking at egg production. And these tortoises in the wild, they live in the Kisapum Desert there, near Okada. The tortoises spend 9 months underground. So the males pop up early because they're waiting for those females to pop up. But in March, the males pop up and they're looking and running around looking for the females. Females pop up a little bit later in March, maybe the beginning of April. But they have 3 months to get everything done. They've got to recover from the last winter. They have to get ready for the next 9 months. And of course, the males are chasing the females around, so they got to get ready for that. But the females also have to get their eggs ready in that short window. So it's a very, very narrow window in which they can get all this accomplished. Now, what I was looking at egg production. And because there's such a narrow window, if you look at their activity and how fast they move around the ground, they just seem like little guys just running all over the place. Especially if there's a male chasing a female or 2 males fighting over a female. Now, if I'm doing X-ray work to look at egg production on the Mojave Desert tortoise, I X-ray them every 2 weeks. But we knew that we need to be careful to plan, because I'm only going over there for one shot for 6 or 7 weeks to get it right. We decided to X-ray them every week. It was the best thing we ever did, because we would have missed probably half the clutches of eggs. They turn over the clutches of eggs every 11 days. And so what happens is, over here in March and April, they're getting ready, but then all of a sudden, boom, they start producing eggs. And it's 70% of the females producing eggs here, 100%, 100%, and then starts to drop off. And they produce 2, sometimes some of the females produce 3 clutches of eggs. Now, this can also be interpreted as being a valuable strategy. One they already have this sort of short window to work in, but if their egg laying is all sort of synchronized, it may not be synchronized, but if it's all within the same narrow window, it can potentially swamp out the predators. Predators have, all of a sudden they've got a whole bunch of nests that they can't find, or they just don't have the time to find. So this is potentially an adaptation that helps them overcome predation on the nest. Okay, let's go on another trip. Let's go to South Africa. I've done a little bit of work in Madagascar, but not necessarily what I'm talking about today. But I spent 5 years in South Africa. A wonderful time. It's a wonderful place. Now, some people call South Africa tourist Mecca. It's got more tortoises than any other country in the world. It's about the size of Texas, but it has 13 species of tortoise. I went there as a postdoc to work with Dr. Hoffmeyer, Major Hoffmeyer. Next slide, please. And we worked on several species while I was there. Mostly on these species here, geometric tortoise. Now, this is arguably the most endangered tortoise in the world. The one in Madagascar, the Untitled, may compete with that species for that title, but it's not necessarily the title that one wants to have. The world's smallest tortoise, this one here, the speckled podlover, lives on the west coast of South Africa. I'll talk a little bit more about that one in just a moment. Close relative to that is what we know here is the parrot-beaked tortoise. In South Africa, it's the common podlover, or the Gewone podlovertjie. Angular tortoise, Roy pants or keel pants, because it has a red or yellow belly, which is, those words are Afrikaans for red or yellow bellies. And tent tortoises. There's different subspecies, and Rita in her program, she's doing morphological genetic analysis, which suggests they've got it all wrong. There's even more. And also the leopard or the mountain tortoise. Now, before the next slide, I'm just quickly mention one reason why I went there first was to do health assessments. reproductive ecology, so looking at eggs and ovarian cycles, nutritional ecology. So those are some of the main things we were doing, especially that first year. We were doing health assessments on 4 species each season, males and females, and some of them at different sites. So we were flat out busy. So one day after the field review, we were complaining to each other that we're just too busy, we're not going to take on any more responsibility. The phone rings. We've got 54 tortoises that have just been confiscated from 2 Czech poachers trying to carry those tortoises into Mozambique. Do you want to do health assessments? Yes. Okay. We were small, but this is one of the main reasons why we were doing health assessments. This was an opportune moment to deal with it. So with the health assessments, we're using something similar to what we do here for the desert tortoise. We look at the upper respiratory disease, we look at the eyes, the skin, see if there's any trauma to the shell or shell disease, things like that. But we also do things, relatively simple things like measure the body mass and express it relative to the care site, and then that's a body condition. But we also take blood samples, hemoglobin, hematocrit, red blood cell counts. In the plasma, we've also sent off to look at various plasma electrolytes and enzymes. So those are the sort of basic things that we do in the health assessments. And we had just been in the field doing this on the geometric tortoise, the angular tortoise, and that Pelvic tortoise. We had fresh data in the field. Those tortoises, we wanted to compare our result to those tortoises. And the poachers claimed that they had just naively walked through the field and found these 54 tortoises in the middle of the dry season. When just the week before, we had 12 people in the field and we couldn't find that many tortoises. So we compared our data to theirs. And in almost every instance, whether it's body condition, like shown here, males and females of the geometric tortoise, but for the other species, but also hematocrit and hemoglobin concentrations. Simple things that you can measure within a few minutes. Almost all of those things, the wild tortoises were healthier than the confiscated tortoises. So what that suggests is that those confiscated tortoises, they weren't just pulled from the field a couple of days before. And probably had been held in captivity by locals who were holding them for those Czech poachers. These data were used in the court case. These data contribute to the body of evidence against those Czech poachers and help convict them. Okay, another sort of cool thing that we did, we had a student, Victor Luhr, who was doing his PhD on the smallest tortoise in the world, speckled Balboa. We knew that they produced big eggs, but we didn't know how big. We also didn't know a few other things about their body morphology. Next slide. But we did know that they were big eggs. And in fact, we measured them. These eggs represent between 6 and 12% of the female's body mass. I hear all the mothers out in the audience groaning as well. And there are a lot of parallels. So this egg obviously cannot go right between here and here, the pelvic canal, if you take it lengthwise. But if you oriented it long in excess, can it still go through here? Next slide. In addition to the egg having to leave through this pelvic canal here, it also has to leave between the end of the plastron and the end of the carapace. So you have the plastron here and the carapace coming down here. The egg has to come out there, and you still have the tail in the way. So and that gap itself is actually narrower than the pelvic canal. So how do these females get this egg out? It's a hard egg. It's not as soft like some turtles and lizards and snakes. It's hard to carry this egg. Well, we know that shell kinesis is important. But it wasn't until we did this study that we know that they also had to do something with that pelvic girdle. We think it's very similar to what happens in humans. And there's actually other vertebrates and invertebrates that use a hormone called relaxin. It helps relax the pelvic canal and the pelvic girdle so that they can help pass the egg. We think, but we haven't tested for relaxin in that species. But we did measure and found that 95% of those eggs are wider than the pelvic canal. Again, it's a hard-shelled egg, that pelvic canal has to give somehow to pass that egg. Next slide. We look back in the literature and there's an anecdotal report. And we'll talk a little bit more later about what can I do to help. This was basically an anecdotal report. This is a more recent photo, but the anecdotal report said this female was laying its egg. Again, I should also point out that they produce only one egg at a time, but it's one bladed egg. That plastron deflects down. That carapace deflects up to help them pass that egg. If you look very, very carefully, and this was reported back in 1976, you can see a little bit of white here and here. That carapace is deflecting so much up that the scutes are separating. You can see the shell in between. That white is the bone of the shell. In this 1976 report, they reported that as well as bleeding occurring at those joints. So I'll have another question I'll leave with you, and we can come back to later. And that is, why? Good question. Why do the females produce only one egg at a time? And there are a few other species that do that in South Africa. Why do they produce such a big egg? They go through all sorts of morphological craziness to expand the pelvic girdle, to deflect the plastron and the carapace, to produce this one big egg. Why? Okay, so that was just one of the many wonderful things that, a couple of wonderful things that we did in South Africa, and we're still collaborating. But when I came back from South Africa, I needed a job. So I actually landed a job with the Marine Corps. The Marine Corps has environmental division, natural resources, and environmental affairs. But the opinions I expressed today are not representative of the government. Or may not be representative. So one of my main responsibilities is to oversee tortoises on the base, the conservation. We have a biological opinion that says the Marines can train here, but you'll do things to minimize the take of tortoises. And one of the most simple things is to tell 40,000 Marines that come through there every year, what is a tortoise? What does it look like? Don't run it over. What happens if you see one? Who do you call? You stop and you call. Or should they call me or one of my coworkers? Just letting them know means that there are 40,000 people on the field with eyes and they can see these things, and they do. So it helps. It helps to protect the tortoises on the base. Well, that's just one of the simplest things that we do. Next slide. We also monitor the populations with surveys. Again, we do health assessments, looking for shell disease, herpes viruses, and primarily upper respiratory tract disease. I was actually on the original health assessments back in the 90s when we were looking at that in the field. We're continuing to do studies on reproductive ecology. And we also monitor animals, individual animals in the field with radio transmitters on them, and we radio track them so we know where they are, when they're active, how far they move. And so those are some of the things that we're doing. Now, one of the reasons why I'm here is because many of you heard about our Head Start program. And so I'll talk a little bit about that now. Now, the species is listed as threatened, okay? And that's because of a multitude of threats, whether it's highways, whether it's military training, trash, ravens, coyotes, disease, poachers, all sorts of things. A whole long list. And the Fish and Wildlife Service has developed what's called a recovery plan. It's a basic plan to try to figure out what's the best way to protect these species and have the population of the species recover. In that plan, they have one section that talks about population augmentation. Can we bolster populations? Head starting is one possible way of doing that. So our installation decided to try this process 11 years ago. We just started, and we built it 10 years ago. I actually got to help pour the concrete and stuff like that. So head starting might be one way to help the species recover, help bolster populations. But it's not been proven. There was a program at Fort Irwin, a program at Edwards Air Force Base, and now there's another program in the Mojave Desert. We might try and advance it on the way out to Las Vegas. Those first two programs are no longer working. They have not determined whether or not head starting can work. But our goal is to determine if head starting can work. With the basic idea, so that means long-term commitment, not just sort of going out for a couple of years and seeing if you can make eggs under these babies. That's not a true test. So the basic idea is that we get eggs from the females. I'll talk more in detail on that. We get eggs from the females that hatch inside of our protective enclosures. And then we protect them from predators, such as ravens and coyotes. So they're protected, and they can grow. And the idea is to grow them up to a certain size so that they are less prone to predators, especially the ravens and the coyotes. And so this last year, September 30th, we released our first set of head start animals. They're 9 years old. Like I said, it's a long-term commitment. Okay, so how do we do this? Well, one thing is we're working in an area where there's a decent population of tortoises. Good habitat, there are tortoises around, we see males, we see females, we do occasionally see juveniles. And so we started putting transmitters on the females. Every month we track them to find out where they are. And I say we, because this is actually a collaboration. That's not, I don't get to spend that much time in the field. I do spend some. But most of this work is done through UCLA. We have collaborators, Dr. Nagy, over here, and Scott Hayward. They have lots of experience doing this. In fact, they've worked at 2 of the previous programs. So they're working with us to help implement our program. So we track these females every month. In the springtime, we use ultrasound and X-ray to determine when and how many eggs they're going to produce. But at that point, we take the females. Okay, the females are not ready to lay eggs within a few weeks, usually 2 to 3 weeks. And we bring them to this facility, which, when you boil it down, it's just a fence. It's relatively simple in concept. It's right in the middle of the tortoise habitat. We fence it off, and we protect the nests and the predators, the eggs and the juveniles from predators. Now, I didn't name it. One of our previous directors named it Tracers, sort of a military acronym. But it also represents tortoise research and captive rearing site. And that term actually indicates 2 of the main functions: tortoise research and captive rearing. Tortoise research helps us not only improve our method of head starting, but also helps us understand the species, so that not only can macaques or the Marine Corps Base 29 homes improve its management, but everyone else. We share that information with other folks. And the captive rearing is just rearing up the animals, head starting them, so that we can hopefully bolster our population on the base. Now, we bring the females in, and as I mentioned, it's basically a fence. It's partially chain link fence. But at the bottom, we have hardware cloth and flashing, and those are buried. Being buried, it keeps out coyotes, kit fox, badgers, and ground squirrels. Ground squirrels can run right through chain link. So we put hardware cloth on down here. Well, ground squirrels can run up hardware cloth. So at the top, we put this metal flashing. And actually, I learned this technique out of the Tortoise Center in Las Vegas when I was working. That keeps out most rodents. So we actually do a pretty good job of keeping out most of the mammalian predators. On top, it's a little hard to see in this one, but a little easier to see here. We have a huge aviary net, and that keeps out the rapids. So basically, it's a fence, but it's basically to protect the babies from the predators. That's the main thing that we do. You'll also see right here, and over here, and here, and here, sprinkler heads. So we have an irrigation system that periodically, not too much, but periodically we turn on the water, the tortoises get a chance to drink. In the wintertime, we really emphasize it, because by irrigating, we can get the native seed bank to germinate. That's what comes up, right, is whatever seeds are out there for the tortoises to eat. So little babies can learn to eat what they're going to eat once they get outside, because that's what's in their habitat. So we're not feeding them tortoise chow. We're trying to get them to eat the native plants that are out there. Okay, so we bring the females in. This is usually in May, sometimes early June. And we bring them in and put each one in its own individual pen. That individual pen has two benefits at least. One is when the hatchlings come up, you know who the mother was. Paternity effects and things like that. But also, by keeping the individual mothers and their hatchlings separate from the other ones, there's less risk of spread of disease. Okay, so then the females come in here, usually in May, maybe early June, 2 to 3 weeks, and they nest. And usually the tortoises are nesting. I mean, all the tortoise nests that we found are actually in burrows. And we type 2 burrows. If they want, they can use those 2 burrows in each of those pens. And or they can dig their own. Most of them use what we give them, but some decide they want their own digs. Sorry for the pun. So the females do that, but the females also have the opportunity to drink and to eat. You know, in a good year, it's not such a big deal. But in all these droughts, usually we've had for the last 4 years, they're coming in and they get a chance to eat and drink, which they would not get in a while. So they're also benefiting from that. Now, female tortoises, desert tortoises, usually show little to no parental care. And I've seen it in the wild, there's a little bit of evidence, but almost no parental care. So we only keep the females here long enough for them to nest, and we put them back. They're up back in their home area, they're all walking around, and some of them actually produce us like a bunch of eggs in the wild. So then we have nests with eggs inside the pens. They're protected from predators. I should add one more thing. We also have a native species of fire ant. And they'll eat the babies and they'll eat the eggs if the density of the ants becomes high enough. So what we do is we put out a little bit of ant bait and knocks down the ant population. Doesn't kill them all, but reduces it so that they don't become predatory. Edwards Air Force Base, their head start program, they unfortunately learned the hard way. 30% of their first batch of babies were killed off by fire ants. I'm sorry, the second batch. The first batch were eaten by ground squirrels, which is why we also have that fence there. So, like I said before, we're all learning. Okay, not everything's working, and who would have known that there's a desert fire ant out there eating tortoises? Just learn. And so what it also tells us is that we can't irrigate too much. We should not irrigate too much, because that will bring back in the ants. So we're conflicting by helping the tortoises, we're also hurting them by bringing in other ants. Okay, so we put the females back, and then we have nests in the pens. So that they hatch. Those eggs hatch, we get hatchlings. Nice cute little babies running around. Okay? Or sleeping in Kansas, one is here. Sky and Ken are out there. I've been out there too, but mostly Sky and Ken are out there counting and putting little labels on these tortoises. So it's, you know which mother it came from and which egg it came from. And we weigh them over time to get a chance to eat and drink, and they grow. And they grow, and they grow, and grow, up to the point, and so last year, they were big enough that we felt it was safe enough to release them to the wild. So we had a little media event where Colonel Park came out and helped us release one of our tortoises, 2-4. 2-4 came from female number 2, and it was her 4th hatchling. So we're monitoring her and 34 others. We're monitoring that hatchling, juvenile hatchling. We're monitoring that juvenile as well as 34 others that we released in September. So September was our first release of 34 of 475 juveniles that we had. So tracers, up to this point, had 475 juveniles. We just released 35 after 9 years. Now after 9 years also, we're already comfortable that we could get babies. That's not hard. I mean, we all know that as well. That's not the hard part. What's more hard, more difficult, is trying to answer these 3 main questions. What happens once you release them to the wild? Do they survive? And how long do they survive? And then, are they still going to become functioning members of the population? Okay? You can have little bodies that go walking around eating food, but if they're not becoming reproductively mature, they're not contributing to the population. So those are the sort of 3 main questions that we're trying to address with the longer-term perspective. You can't do that. I mean, the ones that released at 9 years old, they're not mature yet either. So we need to study them longer to see if they're also contributing to the population, as well as surviving. But along the way, especially during 9 years, we have opportunity to look at other research questions. Questions that might help us understand the species and perhaps help us understand other things to help protect the species. We could not answer this question, vertical transmission of disease. Do the females, if they have upper respiratory disease, infect the babies in the process of producing their eggs? We could not answer that question. That was one of our first questions. We couldn't answer that question because we had essentially no females that were diseased. We had one female that 1 year who had symptoms and tested positive. She didn't test positive again, and she didn't have symptoms next year. So we couldn't really answer that question. Now, for our population, it was good because we have a low incidence of disease. But the idea was that if we have females that are diseased and are producing eggs, and they don't transmit disease vertically, then those females are still viable. They can contribute to the population. So that question is still open. But some of the other questions, I won't go into all of them, but we've also looked at sex ratios. We do have temperature-dependent sex determination, and we actually have a manuscript to review right now on that. Just briefly, first year we had about 65% females. Second year was about 85% females. The third year was like 97% females. I'm like, what are we doing? So we did it. I'm going off on a tangent, so I'm going to take more than 30 minutes. So it's really cool. So we decided that we were going to have one of my colleagues from Australia to come out and do endoscopy. So with endoscopy, they use a little arthroscope or endoscope probe inserted inside the body cavity right here, in front of the hind leg. Look inside, and you can tell, even though they're 1, 2, or 3 years old, you can tell if they're male or female. Now, you know with tortoises, you can't really tell if they're male or female externally until they're big adults. So we were able to do that using endoscopy. The first 3 years is almost all females, like, what's going on? Well, the 4th year, we said, okay, what we'll do is we'll deliberately put out shade cloths over the burrows to try to keep those nests cooler. Now, I think with many of the species that show temperature-dependent sex determination, there's a rule: hot mamas and cool daddies. Okay, so if it's a hot nest, when those eggs are incubated, they'll become mostly females. If it's a cold nest, they'll become mostly males. So we thought that if we cooled down the nest, we might get more males. And sure enough, we did. But it had absolutely nothing to do with our shading those burrows, because we had control nests right next. They had no shading. And they were also males. Well, we started looking at the temperature data for the area. And those 3 years that we had, during that window when those eggs were incubated, tend to be warm. And that 4th year, 2009, it was cooler by a few degrees. And that's enough to make a difference. So what we found is that there's actually annual variation in temperature that influences whether you can more males or females. We also found that in second clutches compared to first clutches, later in the year, more warm, we tend to get more females. Okay, so anyway, so there's lots of things that we can do. One last sort of thing that's kind of relevant to my last slide is potential survivorship. We found that we can get survivorship inside of our facility as high as 96%. That's equivalent to what we see in adults. That type of information is almost impossible to get in a wild. How do you go out and measure and capture the same little 25-gram individual after a year to see if it's still alive? And how much time and energy do you have to do that? Well, we can do that inside of our pets. Now this is different, it's potential survivorship. It gives us an idea of what is a potential survivorship. Okay, so when I spoke to Jenny, she mentioned that one of the things she'd like me to talk about is what can I do? Not just you, but also me. I have a PhD, and so I'm very, very fortunate to be able to do all these things that I've done. Wonderful experiences. But that doesn't mean I know everything. In fact, we learn that as a PhD. It tells you that getting a PhD, it tells you what little you do know. And all of you know a lot of stuff that I don't know. So, just remember, every one of us is able to make a difference, which is sort of the topic of this first bullet. I also want to mention, 50 years ago, my parents, both of them working full-time jobs and raising a family, same time. My parents went out and developed, in Wisconsin, a water sanitation and quality district that had never been done before. And now that lake, Lake Koshkonong, is recovering because individuals went above and beyond to make a difference. We can do that too. So, what we do, what we know, every little bit helps. Continue what we're doing here in the desert, San Diego Turtle Tortoise Society. Okay? We are learning. The more we learn, the more we can do a better job with what we have, and also what we can do to educate others, so that we can all make a difference. I mentioned before about this potential survivorship in juvenile tortoises, something we couldn't measure really in the wild. Did you know that there's still debate on how long the tortoise lives? And it has a lot of consequences. How do you model a population on how well it's going to survive if you don't know how long it lives? Well, we've got these animals in captivity. I've got one at home. I've had him for 32 years. He's at least 60 since I got him as an adult. How long do these things live? That's the type of information that we get. We get that information because we have pets. We know about husbandry, how to take care of these animals. We know a little bit about their nutrition to help them throughout the supply. We do. Scientists do it too, but a lot of them can't do it. They don't have tortoises in their backyard. I've got one in my front yard. He teaches me stuff almost every day. So, what can I do? What can we do? Continue doing what we're doing. Continue learning and educating. Assurance calls. It's another thing that we can do, especially those of us that have pets. An assurance colony or tortoise is essentially like a seed bank. At the moment, the policy is that we don't move these pets to the wild. There may come a time when our pets are extremely valuable, and they will be necessary to be released into the wild. So we keep our animals alive, we keep them healthy. That's our assurance to help protect the species from going extinct. Volunteer. We can volunteer in different ways. I don't know all the different ways. I'd be a fool to say I do. I do know a couple examples. There's the Desert Tortoise Preserve Committee. It relies upon volunteers and a lot. Putting up signs, putting up fences, things like that. That's what the Preserve Committee does. Relies a lot on volunteers. It's a small, non-profit organization with a very focused mission. But that's what makes it very effective. That's what it does. So, that's one way you can volunteer to get involved with the Desert Tortoise Preserve Committee. That's not the only one. That's one I know of. Citizen science. A lot of us want to get involved in citizen science. It's something that's not necessarily a rage, but it's something that's becoming more common. So, we can get involved with some of the researchers. One in particular I know of is actually working with Joshua Tree National Park, Cameron Barrows. He's got volunteers that come out and help them do surveys. They are contributing to our knowledge base, how to understand the species, how well they do in the wild, and how they are responding to climate change.